Hello everyone. Wish you all a very good morning from our team at BITS Technology Enabling Center. Welcome to our series TechMate, where visionary inventors converge to pitch their cutting-edge technologies to students, startups, MSMEs, and industries. With this series, we aim to spotlight faculty innovators from various educational and research institutions, highlighting their innovations across diverse industrial sectors. This platform is designed to forge pathways towards impactful technology transfer and dynamic commercialization opportunities. To elaborate a little more about who we are and what we do, I would like to invite our coordinator, Professor Deepak Chitkara from BITS Technology Enabling Center. So please.
Thank you, Dr. Bhavisha. So the esteemed speakers, Professor Sunil Bhand and Professor Arnab, the representative members, nominees from the partner institutions, faculty colleagues, the students and startup representatives. It's an honor for me to introduce the BITS Technology Enabling Center and what we do over here. So basically the Technology Translation and Innovation Division of the DST has been implementing the technology-related programs quite since long. And the idea is that can we create the entire ecosystem for technology development? Now, when we talk about creating the ecosystem, so there are many stakeholders which are involved in it. We have regional academic institutions, R&D institutions, industries, MSMEs, startups, and the incubators. They all need to bring at one place, and a common platform has to be provided so that there could be certain discussions start happening. Now, one of the major component in this ecosystem is the innovation, and innovation come from the students and the faculties. The technologies which are being, going towards the patent or which are being, patented, and that those technologies has to be taken forward. And that is the whole idea of bringing this TechMate series, where we are identifying the key innovators and creating a where these technologies could be put forward. Now, for any technology to see the light of the day, it has to cross multiple steps. In fact, the multiple steep steps it has to cross, and without partnering, that may not be possible. So, partnering with the industries, industry associations, incubators, IP experts, that is what the goal of this BITS Technology Enabling Center, that can we bring everyone together so that technology translation can happen. So on the academic side, we have partnered with 5 institutions in the Rajasthan, which is Vivekanand Global University, MNIT Jaipur, Central University of Rajasthan, Ajmer, Maharana Pratap University of Agriculture and Technology, Udaipur, and Bikaner Technical University, Bikaner, to achieve the mandate of the BITS-TEC. Apart from that, we have also partnered with various associations such as Rajasthan Chamber of Commerce for various activities. Also, we are interacting with the Ministry of MSME to understand what are the problems which the industries are facing, and also what are the technologies which are available that could be taken up by the industries. And today also, we are going to see these technologies which Professor Sunil Bhand, who will be talking on the breakthrough device which he has created in the lab for quick and reliable mycotoxin detection. And also we'll be looking forward to hear from Professor Arnab Hazra on the methanol testing, the device, the portable and affordable device he had made in the lab, and how we can take that forward. So that is the entire goal of this series and one of the mandate for BITS Technology Enabling Center. So with this background, I thank everyone once again, and we invite you to be a part of this exciting journey, a journey where every step is a leap towards the future, where technology is not just advanced, but is meaningful, where innovation is not just celebrated, but is impactful, and where ideas are not just born, but are empowered to transform the world. So with this, I thank everyone for joining this session and look forward to hear the talk from Professor Sunil Bhan and Professor Arnab Hazra. Thank you. Yeah, over to you, Bhavisha.
Thank you, Deepak sir. In the second episode of our series, as Professor Deepak already said, we feature 2 faculty innovators from 2 different BITS campuses, that is BITS Goa and BITS Pilani. Being educators and researchers for years, these scientists have come up with brilliant innovations tackling some of the most pressing challenges in the field of healthcare and biotechnology. Our first speaker is Professor Sunil Bhand. Professor Bhand is a senior professor and project director at BITS Biosciety Foundation, specializing in fostering innovation and industry-academia collaboration. With 70 publications, multiple patents, and 25 plus funded projects, he's led pioneering work with government, industry, and international partners. Professor Bhand has mentored numerous PhDs and is actively involved in national and international advisory roles. We are very happy to welcome you, sir. It's over to you now.
Thank you, Bhavisha. Am I audible?
Yes, you're audible.
Yeah, can I start? If it's all right.
Sure, please, sir.
Good morning, all, and thank you, TC, for giving this opportunity. Today, I'm presenting about the technology for aflatoxin M1 detection. Primarily focuses on the problem in the country because.
Sir, if you would just, sorry to interrupt, if you would like to switch on your video, you're not visible on the video, sir.
Should be fine now, correct?
Yes,
Yeah.
yes.
So I'm going to talk about a technology which has been developed jointly, with the input of the ministry, the government of India, particularly coming from the dairy industry to the Ministry of Agriculture. We are the world's largest producer of milk. And that's where, coming from industry, the problem statement, this was way back 2016, 15, 16. And we were funded a big project on solving this problem, particularly mycotoxin and pesticide residue. So together with National Dairy Research Institute and Indian Institute of Technology, Delhi, we were partner. I was leading the project. It was a $5 million funding for technology development. So this is one of the technologies which we have developed using that funding. So the first slide, I want to make you aware that while we received the problem statement from ministry, problem still persists today, as you could see on the screen, that very recently, if you see, there are still reports of milk, quality and contamination. You see Telangana, this is a very recent report in 2019 that 50% of milk sold does not meet the standard. If you see, there were reports in Kerala, very recent reports again, 21 and so on, and also 2024. So there is a national report conducted by FSSAI, the Food Safety Agency of India, that still milk problem persists. That's part one within the country for the public health and quality of milk. The other internal input, which I will not be able to share exact number from the ministry, but the exported milk powder rejection rates are in thousands of crores annually. And after sending the material or milk powder from here, they get tested in Europe and elsewhere in the world, and when they are tested, they are not found suitable because not meeting the regulatory standards. And because of that, huge rejection and huge losses, and that's why the economic issues are there, and that's why the country or the government raised that can we have a sensor which is sensitive enough, can be a portable, and can be deployed at the export dock. So this is the problem statement I wanted to share with you people. You see the global scenario also. There's a recent report in New Food from US, wherein the says continent-wide, if you see Africa, Asia, Europe, and Americas, Europe having the highest or most stringent standard, you will see the less, violation or less cases of, exceeding limits, but still 0.2% to 12%. So this is US standard versus European standard. So Africa and Asia, very high levels of rejection, you could see 22.5%. And so also in America, which has 10% relaxed norms against EU standards, so they are also 25% rejections. So opportunity and problem both are huge. So if I look at now the problem statement, India, despite being largest producer and not as exporter, we suffer a lot of economic losses. So our approach was to come up with a solution primarily which can meet the standards of Codex, which is 500 part per trillion or 500.5 microgram per kg of milk. And EU is 0.05. So while US FDA is 0.5, EU is 0.05, 50 part per trillion versus 500 part per trillion. So That's one problem statement. And through our project only, we brought this milk AFM1 limits in the country. It was not existing in 2017. So one major breakthrough was awareness and the need because of the economic losses. What we took an approach is, as you could see on one of the slides, that we developed a biochip, which has been now patented and granted patent both in India and UK. So we have both, internationally and Indian patent granted to us. Indian patent got very much delayed for many reasons. I won't spell out here, but because of fierce competition and interest of the international players. And that's where you could see that you have a chip which is of the size of few millimeter, and that has been, coupled to a portable analyzer, and then you can do analysis within less than 15 minutes. I will come to the other competitive or existing methods, but our technology is indigenous. It has been jointly developed with thin film device, which uses a very low cost material against the market, which uses gold and very expensive materials. We are probably claim we are best in the world in terms of detection limit validated. So minimal sample preparation and high precision, less than 10%. Over the batch-wise analysis, we are not talking of few samples. We have done several batches. Well established for the technology. Yeah, so this gives you a transition of the setup. If you see the leftmost, we started with a very simple wired connector and a transducer, and then you have the manual connector to the chip and so on. And then we moved on to the next connector design, and then from the connector we went to the portable setup, and now we are having our chip getting connected to this portable setup, which is lower corner. And we also developed in-house software using mobile computing input. The data can be sent to an Android device, and any user, it could be end user and individual, a regular traceability, that where from the milk is coming and what was the contamination level. So this is the status of the development of. You could see the comparison. It's very important to validate, with the available technology. So we validated the technology against our patented chip and the commercial kits. I will also share what are the commercial kits available in the market currently. But our chip can do minimum detection of 0.1 nanogram per liter and a big dynamic range up to 1000 part per trillion, which covers right from the Indian standard of 500, USFD 500 PPT to Swiss standard, which is 25, and Australian standard, which is 20. So we are well within the reach of any international standards. As against, you see the best kit from Rita Screen, the Israeli company, is 5 to 80 PPT, and then you could see detection limit, sample analysis time is 15 minutes over 2 hours in a ELISA format, field portable. The other techniques are not field portable. The government or Codex internationally recommended method is Cobra Cell HPLC method, which requires 2.5 hours time and lot of sample preparation, and it is not field deployable. So the certificate for export issuance is a big issue. Many times ship consignments are waiting at the dock for more than 3 weeks, and that's where the economic losses happens, and so also the rejection. So cost of analysis of HPLC is around 4,000 rupees per sample, and when you're doing a big shipment, you have to do thousands of samples. I think that's not economically viable also. So our method can do all the compliances of MRLs, as you could see here, including EU and Swiss ALARA, which is 25 part per trillion. And these are the figures of merit. So what is the current status? The technology is at TRL5. It is being validated with commercial kit. It has also been tested with a couple of milk collection centers and 1 or 2 dairies. We went there and then they used it, and we also used it with the prototype and so on. So that's alpha prototype. So we are expecting to reach TRL7, but at this moment we can easily commercialize the chip itself. Just to mention, there are no currently available biochip for aflatoxin M1. These are mostly kits. The electrode I will also share are not for M1. They are just bare electrode being sold. So we are probably among 1 or 2, few who are into this game. So technology transfer is feasible through licensing board even as of today. And deployment of alpha prototype, we have started now, and we will get the prototype ready, the commercial pre-prototype, within the next 4 to 6 months. And then we take a large-scale trial with FSSI that has been discussed through the NETSCO-FEN that they have through NABL network. It has been planned. And also we have spoken to Nestle, who are ready to do final testing using that prototype. So that's the status. We will be exploring the new patents also. There are a couple of new patents, foreground patents have come out. The funding is important. This was a mega project, and we got a lot of funding because it was an expensive, initiative. So if you look at scenario of the competitors, these are the 3 chips: Metron, Germany, Microx, Germany, and Palms in Finland. And their chips are very expensive. They're not for M1. They're just the bare chip, which people are mostly using for their custom application, but not affordable at all. On the right side, you look at our device, which is a very novel device, very low cost material. Though we have said 200 rupee, but we believe that it will significantly go down in cost when scaled up. So we have technological advancement. All of them you see here are mostly using the gold electrode, where reliability is still okay, but batch to batch reproducibility is still issue. Within this, we have done those analysis and given the feedback, but they are not reliable. The rejection rate is about 30% per batch. So with that aspect, I think we have been having a big technological advancement or these are the 3 kits commercially available. Radar and fixed cost, and also manpower to run. Reagent variability, storage issues are coming despite being the extra cost. So even then, if you compare with that which is currently being used by most of the agencies, either Cobra Cell or the ELISA kit. These are the 2 methods being used by any food safety agency globally. Fine. So that's the technological impact we envisage that it can be deployed for quality assurance and compliance of regulatory norms at milk processing plant, as well as the sensor can be networked for food traceability across a set of dairies or a set of collection points or any of the supply chain. That's something which the government has also intended to adopt if we demonstrate successfully. So also we have spoken to 2 or 3 dairies, the global large dairies like Nestle, Amul, and Mother Dairy. They have shown keen interest because the cost incurred per liter is extremely high for aflatoxin M1 related testing. So this technology will 40 crore people. So if we are able to deploy good quality testing within country, it will also help, and so also the export promotion will increase due to the good quality control. So only the good milk can go for processing of powder. Now let me come to the market size and competitive landscape. So if you look at, I'm talking about, because this comes under electrochemical biosensor domain. Ideally, the global market is, as you see from 2023 report, is about $28.9 billion. And within that, if you look at only the US market, the CAGR projected up to 2030 is 1%. So very, high potential, particularly if you see from the right side bar chart. Electrochemical biosensor hold almost 75 to 80% share. So out of $28 billion within global biosensor market, 75 to 80% is by the electrochemical biosensor. So that's one important perspective. Let me go to the next slide. If you look at another report where the global market has been also projected, similarly growth rate is around 7.5 CAGR. This is another report it's saying, and it also shows the largest segment is electrochemical biosensor. And if you see, US, Canada has been the largest market so far, but also the Asia is emerging market for the biosensor. We could see here in the report that while largest has been so far, Americas and Canada, but India and China are the fastest growing market as per another report from the SkyQuest. So if you look at Indian biosensor market potential in millions of dollars, I think here also if you notice that against the global CAGR, the Indian market is expected to grow at 11%. And that gives an interesting, opportunity to invest into the market and do the Make in India. That's what is the right timing with the semiconductor era entering in India, fabrication technologies and facilities getting up already. I think this kind of thin film devices can be made easily within the country now. So we have a good market potential. These are the companies which are into market. Abbott, which is also itself located in Goa, the facility for manufacturing, and Roche, some of the big players, Medtronic, Johnson & Johnson, and GE Healthcare. I think they are into the game, and we have been keeping a close eye. We have been scouted by one or two of them. I won't disclose the name here, but two major giants have scouted to our technology, including. So there's immense interest due to the game-changing material and the miniaturized sensor. It gives us a lot of confidence that the technology has potential for marketing and commercialization. So if you look at the market drivers, growing demand for, I will not use the word point of care, is mostly used in the report for the healthcare, but for agri food and food safety, I think the market driver is at both the individual end as well as the organized sector. Also, the opportunity is, as I said, is due to the semiconductor and nanotechnology, and also enhancement in the biochemistry perspective, the new receptors being available in the market, the more specific receptor being available in the market. If you look at the constraint, I think for health, it may be a good point to say it has high cost, but that's where we have pitched in to reduce the cost significantly, maintaining the highest sensitivity so far in the market. It has been cited in the World Mycotoxin Journal. It's not our statement. So it endorses that we are much ahead in terms of the regulatory limits as well as meeting the cost affordability. Legal challenges for food diagnostics are not as stringent as against healthcare sector, where the compliances and regulatory approvals are very long. So I think in this case we just need a couple of simple regulatory approvals. One is the license to manufacture, and also if you want to go European market, maybe CE marking and so on. And those may take 6 to 8 months. I don't foresee major challenge in terms of regulatory perspective. If my last slide perhaps, or maybe last but one, the product is a portable biochip sensor for detecting aflatoxin. Sorry, it should be aflatoxin M1 in milk. Also, milk product we have tested, and it can be deployed for field surveillance as well as routine analysis at home or industry. And the major advantage is less than 15 minutes, very low cost. If I may say so, one of the major player in dairy today spends 12 rupee per liter just for aflatoxin M1 test. And that's huge, and that is passed on to the customer. So if I take, for example, a good brand with 78 rupee per liter, I am paying, the customer is paying 12 rupees just for AFM1 test. I think it gives enough indicator for market potential. Ease of use, because you just have a handheld device and a chip like a glucometer, but probably much simpler than that. And we will provide a calibration support if the market is established. So primary market, as I have already shared, large dairies, global dairies, even Dairy Development Board. I have been talked with each of those clients, whatever I have mentioned here. I have been approached by Nestle, even from Switzerland, directly from their technology division. Apida has been in direct contact, and they are ready to do the large scale validation, at least with 1,000 samples that we are already pushing. Food regulators, I am part of a FSSAI committee, and there's a lot of interest shown by the FSSAI committee that if we are launching this through commercial entity or through a startup, they will be ready to deploy. Also, small organized dairy farms. We have been talking to Chitle Dairy, who owns now almost 1,500 cows and buffaloes, and also some other dairy in Goa, which are almost 2,000 animals and at least 10,000 liters per day production. So a lot of interest has been established, and we would like to go for the next step quickly. So the last slide is that market entry. R&D and testing we have completed. Even the batch validation, intra-batch, inter-batch, even chip has been fabricated in 6 to 7 batches, though done at in-house at IIT Delhi Care and BITS. And prototyping, as I said, the pre-commercial prototype already we have engaged with one of the vendors, and it should be ready. I have taken the conservative number. If we speed up 4 to 6 months could be the prototype in our hands. And then will require probably 4 to 6 months for the market, Indian market approval. And if we start production, scaling may take 18 months. So with that, I thank you for your patient hearing. If there is questions, I will be happy to respond.
Thank you very much, Professor Sunil, for your insightful talk and sharing your innovation. Now the session is open for discussion. If anybody would like to question, please write in your chat box so we can put forth your questions to Professor.
Good morning, Professor Bhand. I have a question. Am I audible?
Yeah, you're audible, Mr. Anish.
Yeah.
Good morning.
Professor Bhand, thanks for such a wonderful presentation, and nice to know that the technology is already at a very advanced stage and some of the regulatory aspect has already been cleared and seeked for. One thing which I wanted to know, will it require a customized version for an industry to look into it? Right now it's an handheld device, but for the industry to do detection on a larger scale, will it require a different kind of version and customization?
If you look at a large dairy plant, I think they will need a slightly more number of samples per day. And we already have another version of the chip where 4 channels are available. And that could be very easily replicated. When we spoke to the vendor, we are keeping that option ready, where somebody can opt for a single channel versus one can opt for a multi-channel system. And one can do easily, because since it is only 15 minutes, I think as against what they are currently doing, the ELISA test versus this one, I think we can meet or even exceed the throughput required. So that's the feedback from 2 dairies I have. So some customization will be needed, yes. Answer is that.
Okay. And what will be the customer acquisition cost in case it is sold to individuals for personal testing or a small level testing?
I will not have immediate exact number, but if I look at the analyzer, we are trying to right now go with a commercially available analyzer, but we are also in talk with one of the company for white label, branding for us and lower cost. So end of the day, target is to go for about 8 to 10,000, one-time fixed cost. And the chip today, there are no commercial chip, but the chips available are only for, other users and people use in-house. So I think the chip should be targeted around 20 to 25 rupees per use. If you look at diabetic chip today, the Roche costs 30 rupees per chip. Even though they have rejection rate of about 25% and they still claim on their brand that we don't claim it for medical use. It will be very surprising though it is deployed in the country. So in principle, we are targeting to be much cheaper because they are still using gold electrodes. And that's in one of the version. Now there are 2 versions. One is the spectrophotometer version, another is the electrochemical version.
Okay, sure. Thank you.
So there's a question in the chat box. So Ajay Singh Hada wants to know that does this sensor only work with milk?
Answer is no. We have developed various applications, and since the main purpose initially was to develop for milk, we have done yogurt, we have done flavored milk, we have done even paneer and cheese, and that's one line milk product. We also have now filed 2 patents, Mr. Rajesh knows, from BITS, wherein we are coming up on pathogen detection for E. coli. We already have filed a patent, and Salmonella we are going to file a patent very shortly. So this is a platform chip which can be tailored to at least 2 or 3 major applications. One other area which has come from MPDA, the Marine Product Export Development Agency, we are currently getting funding from DBT is to develop for 2 critical antibiotics used in the prawn or shrimp sector. So it can be adopted. The platform can be adopted. Answer is that.
Thank you so much, sir. And there's a follow-up question. Can't we use it with water? Let's say with food after using a mixer grinder.
Can be done. It's even easier. I mean, milk has fats and other things. Water is the easiest matrix.
Then we have Dr. Sunita Bishnoi. She would like to know about the selectivity of the proposed sensor.
So I think the selectivity, specificity have been very well established. It is only selective to M1. We have checked with M2, G1, G2, B1, B2, which are the analogs for the other total aflatoxin globally. So while some of the countries like EU has given special for M1 and B1 separately, so also US, but Australia gives total aflatoxin. So there are different norms and we could calibrate. So this one is highly selective.
Okay. A follow-up question. Can it be used for human body fluids like blood?
We are not tested. Could be, but that's a separate, optimization needs to be done. It's a platform technology. As I said, it's an electrochemical chip. And if you can tailor make your receptor, I think I don't want to speculate, but there are some optimization. With some optimization, answer should be yes.
Okay. Since you have very nicely elaborated the technology, there is a question which asks, is there any waiting time list in case we want to buy the setup? What will be the price for this setup?
So if I may say so, what can be done immediate today, what we are trying with some of the major players is to license the technology to fabricator. The chip fabrication can be directly done and that could come immediate. And then for the second part, which is the biochip development, we are in dialogue with a large manufacturing firm in Goa who have been into healthcare sector, but their setup can be adopted to automated, production of chip. So as somebody asked for the blood, the same technology and can be easily adopted to the technology, production of the product, the biochip. So we are in dialogue with them and feasibility is high. I don't foresee any major hurdle because theoretically the process industry steps are simple, reagent, dispensing, covering the reagent, little bit of packaging, and that's it. I don't foresee a major difference against a blood chip versus this.
Sure, sir.
So timeline wise for, biochip production, I think the setup adoption, it can be even outsourced. In that case, 4 to 6 months, ideally. You have to optimize the batch production and the costs. And if that is done, I believe should be doable.
Okay. Thank you.
Thank you very much, Professor Sunil Bhand, for your answers to our curious questions. Moving forward with our next speaker in TechMate series, we have Professor Arnab Hazra. Professor Hazra is an associate professor at BITS Pilani, is a specialist in nanomaterial-based chemical sensors with 60 plus research publications. He is a recipient of prestigious awards like Indian National Academy of Engineering Best PhD Thesis Award and Institution of Engineers Young Engineer Award. He has collaborated globally, including Tel Aviv University and the University of Brescia. His research focuses on advanced gas sensors and semiconductor devices. Let's hear more from him. Over to you, Arnab sir.
Yeah, thank you. Am I audible and visible also?
Yes.
Okay. I think screen is also visible.
Yes, yes.
Okay. Thanks to the organizing committee for giving me the opportunity to discuss my invention here. Myself, Arnab Hazra, I'm from the Department of Electrical and Electronics Engineering. So in this discussion, I would like to describe my recent invention for rapid detection of methanol poisoning. It's a device for methanol contamination detection in alcoholic beverages. Here you can see that what is methanol poisoning. If we can say that uh, in different industries or some local producer, they are producing different traditionally fermented alcohols, and due to inadequate distillation, methanols are unintentionally added with ethanol. And that is the problem, because after consuming that contaminated alcoholic beverages, methanol decomposes into toxic formaldehyde and formic acid, which causes health problems like headache, fatigue, nausea, blood pressure, and even death. WHO reported that 500 mg per liter for methanol concentration in blood is in toxicity, whereas 1500 to 2000 mg per liter can be cause of death. So this type of methanol poisoning incident is very common in India, and mostly the poor income people are the sufferers. Here I have presented one image of the incident in June 2024, where the methanol poisoning happened in Karunapuram, and this is the situation. But this is not the only incident. This is one of the recent incidents. If you can see this list, I have listed few significant incidents from 2009 to 2019. These are the only significant thing happened in wide different states of India, and these are the number of deaths. These are the only few significant cases. Other than that, the methanol contamination is a very common problem, and lot of other cases are registered in India. So this is our motivation and background to work with a methanol poisoning detector. And if we can see the recent scenario, the most common equipment for this type of methanol poisoning detection is a chromatography system. So the challenge is actually, if we can say this methanol contamination, we are detecting in ethanol, and methanol and ethanol both are very similar types of alcoholic agent. So if one sensor can detect methanol, it can also detect ethanol. That is simply says the selectivity issue. So in this scenario, of course, chromatography system is the easiest and most convenient way. You can see this gas or liquid chromatography unit installed in our lab, and we can also analyze our sample in liquid phase injection by using auto sampler. But and this is the typical results in spectrum form, and we can successfully isolate the different volatile organic compounds in a mixture. But if we can see this model and the cost, it is around 1.6 crore now we have invested to install it. Okay, and the minimum processing time is 40 minutes. Additional time for expertise and to standardize this system for particular application, or more specifically for the methanol contamination detection. So the system is highly that cost very costly system and time consuming, and expertise should be required. This is another motivation to us to develop one portable and simple rapid methanol contamination detector system. And with this motivation, this is our uh, technology what we have developed for portable and rapid methanol detection. So I have summarized here the specification of this system. This is a battery-driven portable system. It can detect methanol contamination in alcoholic beverages or ethanol within 1 minute time duration. The maximum time will be required as 1 minute. This sensor need no external temperature. It can work at room temperature, and this is very fast device. It can recover within 10 to 30-second time duration. So the sensor will be ready for the next measurement within 30 seconds of its first measurement. Of course, it's very low cost and simple to handle. You can see the schematic of this device have four principal parts. We have a sine wave generator, battery section, signal processing circuit, and this is the sensor, which is the most novel part of our invention. So this sensor is placed inside a bottle, or it can be placed above the bottle, where the vapor of these contaminated alcohols will be available. Now, this invention is actually, we have patented very recently. So a lot of work are still pending for marketing and some commercialization. So this is our patent we have recently obtained, and we have published one paper in IEEE transaction on instrumentation and measurement in 2020. So this invention was happened around four years back. So I also want to acknowledge my first PhD student, Dr. Pratik Bindra. Actually, this is our joint work. So now I will move to the more detailing of this invention, and firstly I will describe this sensor, what we have used for this particular technology. It's a capacitive gas sensor, which is far different as compared to the conventionally available conductometric sensor. Of course, the structure of the sensor is very, very simple and only a three-layer stack, you can say. You can see the side view of this sensor, which is placed on a glass substrate, and the bottom electrode is a titanium substrate, and we have titanium dioxide nanotubes. This is a 1D material is aligned here, and we have a top layer of gold. So this is the simple structure, and if you can see, this is a typically parallel plate capacitive structure. This is the top view of this sensor, and if we want to see this particular sensing material, which is titanium dioxide nanotube, this is a typically 1D structure. You can see these images in scanning electron micrograph, that is a nanoscale, falling in the nanoscale regime, and this is the length as 2.8 micron. Now I can show you the fabricated sensor. These are looking like this, very simple and small structured device. The three layers are clearly visible here. The bottom portion is a titanium, and the dark portion is indicating the nanotubes, and the top portion is the gold electrode. Now you can see the fabrication steps. Of course, multiple fabrication steps will be involved here: the anodic oxidation, thermal annealing, acid etching, patterning and masking, physical vapor deposition, electrical contact formation, etc. So, of course, if we can see these fabrication steps, these are complex and costly, but after following one-time fabrication step, we can develop around 30 to 40 devices. And in that case, the fabrication cost will be reduced significantly. So these individual capacitive sensors have a significant role for different type of other substance detection. Now, I will show you the second part of our invention, that is signal processing circuit. We have, of course, less novelty in this part, but this signal processing part is typically developed for a capacitive sensor. So the configuration is different, and we need a sine generated circuit to operate to apply a input for this particular sensor. So this is the typical flow diagram for this signal processing circuit what we developed, and this is the circuit in simulator. In the right side, you can see the circuit what we developed in the breadboard structure, and the sensing chamber, we have this, and we can place sensor inside this chamber, as I have mentioned in our schematic. And of course, the contaminated solvent, contaminated beverages can be placed inside the bottle. Small quantity will be fine for the testing. We have used simple LED here, 3 LEDs, which will glow at different contamination condition of the target solvent. Other than these LEDs, it can be represented with a display screen for more accurate representation of the methanol contamination. So now I can show you the operation of this invention. And so I can share this video so that you can see that how this device can work and how rapidly it can sense. I think the video is visible.
Yes, it is.
Yeah, okay. So I can skip it and can come to the main point here. So see this structure. Yeah. So we have taken four different bottles of this contaminated alcohols, that ethanol and methanol mixture. I can show you here. Just yeah. So this is a pure methanol. This is pure ethanol, and we have 10% and 20% contaminated solution. We are representing this sensor voltage, which is actually show you that how rapidly the sensor can respond and recover. And this is our timer to represent that exactly how fast the device is responding. And we will show this LED 1, which will glow at 10% contamination, LED 2 will glow at 25% contamination, and LED 3 will glow at 100% contamination. And every measurement will be performed within 1 minute of time duration. So now we can start this measurement, and you can see that sensor is placed in an ethanol bottle where no methanol is there. So now you can see this change of baseline of this sensor is rapidly changing. And of course, for this measurement criteria, we have to wait for 1 minute. So we will see the LEDs are not glowing at all. And because the baseline change is not sufficient. So here is 24 seconds. We can wait for 1 minute. I can skip this so that it will be faster. So after 56 seconds, it is no change. So we remove this, and the thing is very important. Our sensor is recovering within 10 seconds, and now it is ready for the next measurement. And this sensor is without any external temperature device, so it can be used directly to the chamber. It can be placed inside the chamber. This is for the next testing, and we have taken the 10% methanol contaminated sample, and we are placing the sensor inside it. And now you can see the baseline is changing rapidly, and within a few seconds, we will see the LED 1 will glow. As this is the concentration we are using, that is relatively less, so it is taking some time, but result is continuously changing. So we can measure any contamination in between 0 to 10%. So first LED is blown, and it takes 50-second time around. I can move to the next level where the 25% contamination we will see. And of course, this measurement will be rapid. Oh, sorry, I have lost it. This is the second case where you can see the 15 second is the time, and next measurement and next LED will glow within. Yeah, so in that way, I'm not continuing it. When we will see this fully contaminated solvent, then it will be quite faster. But all the time, we will not cross the time of 1 minute duration. Okay, so I can back to the presentation. So now the technology, what we have actually used here for this measurement. So our sensor is the most novel part, and you can see the change of this capacitance with time, and it's a spike-like response, and within a few seconds it is responding. Now the technology what we have used, that is quite different as compared to the normally available technology, you can see that our device is a parallel plate capacitor and with a porous layer of sensing layer, and that is in between two electrodes. And if we can consider the air inside these porous nanotubes, it can show the dielectric constant of only this sensing material. But the dielectric will be significantly differ when the volatile organic compounds can enter into it. So in our case, the volatile organic compounds are the ethanol and methanol, which have quite a high dielectric constant if we can compare it with air. If we can consider the same thing in air ambient again, so due to removal of the volatile organic compounds from it, so it will back to its original conditions and the thing is very rapid. So now the fact that how we can able to detect ethanol and methanol, we can isolate them because methanol have quite high dielectric constant as compared to ethanol, and that is the key point for this particular sensor. So by using this dielectric property and by using these one-dimensional porous nanostructures of a another dielectric material, we developed this sensor and we have patented this application. So this is our overall invention. And now we are working with the PCB development of this technology. And we are also planning to launch this device for different marketing strategies. But the thing is, this particular work is patented very recently. So few work is still left. We can say we are at the position of TRL 4 level. So with this discussion, I can say that this is our inventions. I have added few more slides regarding the some applications and innovation perspective of chemical sensors based on my 12, 13 years experience in this area. So if you can classify these chemical sensors, they're basically classified as gas sensor, electrochemical sensors, optical sensors, and biosensors. So electrochemical sensors example is already given by Professor Sunil Bhand. And this is the typical example of gas sensors having lot of different application area. So I would like to show you different applications and opportunities where people can develop different types of gas and volatile organic compound detection sensors. It have lot of market acceptability. We can consider the environmental monitoring application, where we can see that continuous requirement of detection of different toxic gases and volatile organic compounds need to be detected. Detection will be required by different gas or volatile organic compound sensors. So here is a huge market of that. Healthcare and medical diagnostic, we are particularly working at that area also, because if you can see the breath analysis, that is a big market for gas and VOC sensor. We can diagnose different types of uh, disease in early stage by detecting the biomarker or breath marker in breath in exhale breath of human. So diabetes, asthma, chronic kidney disease, these are the different disease we can detect in early stage by using different types of gas sensors and VOC sensor, and that can be considered for the healthcare and medical diagnostic application. Industrial process, we can detect different types of leakage of toxic gases and vapor. We can consider the food safety and quality control applications in cold storage. We can measure the different stage of uh, 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 the freshness of different foods. We can measure by using gas and VOC sensors. In defense and security sector, we can use sensors for detecting the different VOCs which can release from the explosive, and we can use sensors for soil quality monitoring in agriculture applications. So these are the wide applications and opportunities where we can use gas sensors and chemical sensors. And here I want to show you the market trends and outlooks. Of course, chemical sensor industry is experiencing rapid growth and driven by increasing demand for real-time monitoring and the need for improved safety and environmental regulations. But the thing is that it's not the market says we have to develop a complete portable device, portable detector. We can develop different component of different stages and that have quite valuable market acceptance. You can see here I have shown one flow to represent that portable detector and a packaged sensors on PCB. There is a packaged sensor itself, a single sensor and the sensing material. So market have the requirement of each and every stages. This portable detector, of course, need lot of expertise from materials, expertise from electronics, and lot of other involvement should be required. But these package sensors, of course, these PCB based sensors here, of course, required expertise will be less. And you can see that MQ sensor that is quite available in market for smoke detection and air quality monitoring. Package sensors is now frequently available by TGS and other companies for detection of acetone and NO, different types of gases. So individual package sensors have a significant market. Other than that packaging, we can only develop the sensors in a single component and that have also market requirement. Even the sensing materials can also be separately developed and it can be considered for different marketing purpose. But the market demand specify this two, three very important points. Now, recent time, if we want to focus on the portable detector, so wireless thing is one of the most attractive options. The implementations of IoT is also very important area now from the market perspective. And the sensors have significant problem with selectivity. That is a very important problem to overcome, because if we want to detect hydrogen by using a sensor, but that also detecting ammonia, so that's not a very good thing. So the selectivity, sensitivity, as well as the repeatability will be the important thing, and that is key challenge for gas sensor market. And finally, the collaborations and partnerships. Of course, this flow says that we need lot of collaborations to develop a device, a portable and detector. So these are the overall thing for my discussion, and I would like to conclude here. Thanks to all. Yeah.
Thank you very much, Professor Hazra, for such an insightful talk, not only about your innovation, but also telling us about the various opportunities available in biosensor development. Now the session is open for discussion. If the audience would like to post their questions in the chat box, we can take them one by one.
Sure.
Professor Hazra, good morning. Rashni is this side.
Yeah.
Thank you for such a wonderful presentation and a very strong use case of the technology, which I can foresee. Can you please let us know at what level right now the technology has been validated?
Technology is in, you can say, TRL 3 or 4 level, you can say, because actually in the current stage, we are looking for the PCB design of our circuit. And actually, we have the expertise on the sensor development. So sensor fabrication part actually we do in our own laboratory. And so the PCB part is the pending thing. And after combining these two, we will be in the position for the packaged device.
Okay, sure. And what are those use cases that you have explored with the existing technology?
What are the other possibility to use this technology?
What are those practical use cases that you have explored?
Okay, we have explored it. It can be. We have already tested it with different contaminated beverages. So we collected it from different source, sometimes from market, and we tested our device and before reporting it. So the reports I have not added. So it can successfully detect the contaminations of methanol in any different types of beverages, because the example what I'm showing, that is methanol is only added with ethanol. But if we can take any other real-time alcoholic beverages, so there will be some other compound will be added, because some essence, sweetness, and lot of other things. But in presence of that, this sensor can detect methanol contaminations that we have tested with multiple samples. Around 4 or 5 samples we have tested, and it's working well.
Okay. So apart from the beverage industry, what could be the other industries that can use such kind of?
Basically, this particular device is based on a novel technique, that is dielectric change technique. So where the target analytes are high in concentration and have quite a high dielectric value, so this sensor can be used. I can give you some example. It can be used for humidity detection related thing, because humidity has quite a high dielectric constant. And other than different volatile organic compounds which have relatively high relative permittivity, we can use it for different applications. But if the concentration level is low in lower ppm or ppb level, this sensor will not work.
Okay, sure. Thank you.
So there's a question from Dr. Sunita Bishnoi. She wants to know if these sensors, she writes, we want to use these sensors for detection of a particular compound. So how
Yeah.
interference by similar compounds be eliminated? By similar meaning having almost same dielectric values.
Yeah. So of course, it is application specific. Your question is about the selectivity of the sensor. Selectivity is a situation, it depends on the application also. If this is the particular application, that is detection of methanol contamination, so the maximum interference by ethanol itself. Okay, so this sensor can counter that situation. If any other interfering substance can presence, okay, so we have to optimize, we have to check the change of the sensor performance, and accordingly we have to process in our signal conditioning circuit. So for particular application, I think selectivity will not be the big issue, because our target is for this particular sensor is methanol, which have very high dielectric constant, commonly available as compared to commonly available any other volatile organic compounds. Selectivity situations we have tested, but that is not the big issue here. And in advanced stage, if you want to know that how we can overcome for a general situation, so the multiple array of sensors will be the solution. And in that case, by using machine learning, one can achieve selectivity. So that is the most common technology used for any electronic nodes.
Okay. Will this sensor work with any fermented liquid?
Of course, it can be useful for any fermented liquid. Of course, we have tested it.
Professor, I had a question. Are you looking at any particular commercialization strategy? I know it is still at a low TRL level, but once it is quite mature, are you looking at the commercialization part in a specific way?
Yeah, we are looking at for because the patenting was taken some time, and now we are basically mainly looking for the PCB part of this sensor circuit, and rest of the thing, that fabrication steps we have already developed, and that flow is well established in our lab. So that is a part is painting, and then we are also talking with our some TVI team for the startup with my students, my PhD scholars. So they are planning to commercialize it very soon. But not only this is a device, we are also working with individual sensors and sensor related components for commercializing, because portable device, its marketability is sometimes we have seen that is challenging, but individual sensor components, sensor related and different accessories, which we have a huge expertise. So we are also planning to commercialize those things.
Do we have any other question? Thank you, Professor Hazra, for your answers to our questions and also such a wonderful, fantastic presentation. At this point, I would like to thank both our speakers today, Professor Sunil Bhand as well as Professor Arnab Hazra, for their insightful talks and sharing their innovations with us. Before we conclude today's session, we would like to give you a glimpse of Technology Enabling Center at BITS Pilani, our resources and services, which can be helpful to everyone in their entrepreneurial journey. So I'm inviting our IP and Technology Officer, Mr. Sahib Sheikh, to elaborate more on this.
Yeah, thank you, Dr. Bhavisha, for elaborating this point. Hi everyone. So this is our BITS Technology Enabling Center, supported by Department of Science and Technology, and coordinated by the, Professor Deepak Chitkara, and co-coordinated by Rajneesh Kumar, head of TTO. So this is we and our team. The resources that we have developed till now are TRL calculator and IRL calculator. Basically, you have seen that entire presentation, Professor Sunil Bhand and Professor Hazra was talking about the technology and that technology readiness level, validation level. So if you are from any innovator point of view, want to validate, use this technology readiness level calculator, you just have to go in our web page, and there are calculator tab is there, and you can access this. Basically, you have to answer some questions in a yes no format, and the calculator will assess your technology readiness level. In the same way, there is investment readiness level also there. So to assess the investment, how much in IRL calculator IRL level is there for your technology, and the business potential of your technology, you can access this investment readiness level calculator. And next, the most two important dashboards and resource that we have mapped till now. One is Rajasthan Expert Database. As you can see, this is basically, we have collected all the experts from different institutions of the state of Rajasthan, academic institutions, and we have mapped the expertise. Like you can just search in our dashboard key, who are the expert in the particular electronic sensor domain, and you will get all the experts from different academic institution, and you can access them. You can connect with them for further research project, further collaboration, or any technical help if you need. And there's another important database that we have created, that is Rajasthan IP dashboard. As you can see here, basically we have mapped all the patented technologies in the state of Rajasthan. So you can see in the state of Rajasthan, there's roughly there are 1,500 technologies available in different technology segment. If you need, if you are from MSME or if you are from a startup background, you're struggling in any kind of technological solution to find, you can come in this dashboard and you can search in that particular segment what are the technologies available in the state of Rajasthan, and then you can reach out to us also for further commercialization, or if you want to take that technology, if you want to scale up that technology for your industry, for your company. So these are the resources right now available and live on our website. You can go www.BITS-TEC.com, and you can visit these resources that we have created. And lastly, we also provide the IP support. As you have seen the entire journey of these two innovators, they have filed a couple of patents regarding their technology. So basically, it's very important to protect your technology via IP. IP acts as a shield for your technology. So if you are from any of the stakeholder from MSME startup or a student or innovator, you need any kind of IP support from us, you can reach out to us regarding patent, design, trademark. Please do contact here. You can scan this and you can write us to here. So basically your technology and your idea will be valued. So do not hesitate to contact us. Please write us in that given mail. So thank you. If you have any question, you can put in the chat box. Otherwise, over to you, Avasha ma'am.
Thank you very much, Sahil, for apprising our audience about Technology Enabling Center at BITS Pilani, our activities and our resources. Now I will invite our co-coordinator, Mr. Rajneesh Kumar, to give a vote of thanks.
Thank you, Bhavisha. Such a wonderful engagement today with all of you. Specifically, I would like to thank Professor Bhand for showcasing his technology and the TRL level that it has been reached. And it's really nice to know the way the kind of new technologies are being developed at BITS Pilani in terms of their readiness, in terms of looking into the market acceptability, in terms of the market problem that we are solving at our end. I would also like to thank Professor Arnab Hazra for the development of its technology for methanol detection, which I foresee as one of the promising technology, because there are very different and unique use cases that can be explored with this technology. Above all, I would also like to thank our participants today for engaging with us to listen to some of the technologies which we are developing at BITS Pilani. We are open to engagement to any of with our participants who are interested to talk to us with respect to any of the technologies that we have developed. It could be a joint collaboration for research and development. We can also explore technology licensing possibility with you. Thank you for your time for attending this session today. I would also like to thank our leadership at BITS Pilani, who's promoting innovation at all levels, from a student to faculty, to take this innovation to the market. Specialized units has been created. We have a specialized IP cell that takes care of filing all the IP applications. We have tech transfer office that looks into commercialization. We have BITS-TEC enabling center that has been supported by Department of Science and Technology, that totally accelerates the innovation that is happening in the region of Rajasthan specifically. But in general, we are open to support any of the innovators, startups and MSMEs, those who are there and those who are looking for any of the support in terms of taking their innovation from lab to market. As Mr. Sahil has shown you the different services that we offer at our platform, please do reach out to us, and we will love to explore those possibilities and would like to work with you to take you to the next level, to take your innovations to the market itself. So with this, I would like to thank everyone for your time today, and we look forward to your participation in our future workshops and programs. Thank you all.